All right. Hi, everybody. We are going to talk about two things today that are found on your Unit 6 learning targets that are super out of place in your Unit 6 learning targets. They don't fit in the chapters where we're reading, but the College Board has them there. So I'm just going to do a presentation on the topics, and you just won't be doing any reading. They should be familiar because you've heard kind of these ideas already, but we need to really make sure that you feel solid about the concepts. So the first one that they ask about that we haven't covered is something that's called horizontal gene transfer. It's basically how prokaryotes are able to evolve. So we're going to talk about how they can exchange DNA with each other. And then the second thing we're going to talk about are retroviruses, and they are really retro. No, retroviruses, HIV, for example, is a retrovirus. And so we'll talk about retroviruses and what's special about retroviruses. Okay, so right off the bat, how do prokaryotes evolve? Bacteria clone themselves in the process of binary fission. That's their form of asexual reproduction. It's very similar to mitosis, but it produces clones. So if they're cloned, how can they evolve? How can they change over time? How can they develop antibiotic resistance? Since they don't have sex, bacteria don't have official male and females, they're all identical. And sex would increase variation. They have evolved alternative ways to exchange DNA with other bacteria, and that's what we're going to call horizontal transfer or horizontal gene transfer. There are 4 ways. You should write down all 4 ways that the bacteria are able to do this. And the first one is shown in this image here. The first one, give me a second. The first one is called transformation. We use transformation for genetic engineering experiments, but transformation has been around for as long as bacteria have been around. Transformation is when a bacteria is able to take DNA from the environment. What? Why would DNA ever be in the environment? From dead bacteria. So bacteria that have died for whatever reason, their guts are spewed all over the environment. Other bacteria can come and suck up their DNA, and whatever that DNA had in it, now that bacteria possesses it. So for example, if this happened in human beings and I saw this like amazing, let's say Tiger Woods died in front of me, I could walk up to Tiger Woods and I could suck up his DNA, and then suddenly I'd be this amazing golfer. That's what these bacteria can do. So that's called transformation. Now, we use transformation in genetic engineering experiments where we genetically engineer a plasmid and just throw it into the environment with bacteria. There's a little bit more to that. We can force them to suck up a plasmid, but then the plasmid, and they're showing this in letter B here, the plasmid can be taken up into the bacterial DNA and into the bacteria is what I mean to say. So there's transformation with DNA fragments and transformation with a plasmid. Next, number 2, again, we're talking about how bacteria can get access to new DNA, which allows them to change and evolve. The second way is called conjugation, and conjugation is sometimes referred to as sex, but bacteria don't have male and female, so this is not sexual reproduction. The bacteria can actually exchange genetic material through an organelle. I don't know if organelle is the right word. Through a projection that grows off of some bacteria that's called a pilus or a pili. And you can see where it says conjugation tube in the image. That's where the pili have come together. And they can transfer DNA from one bacteria to another. They can even do it between species. So they don't even need to be the same species of bacteria. They can just trade DNA that way. So that's one way. And it's kind of like sex, but they're not male and female. So it's not really. All right, the third way that bacteria can get new genetic information. Now, this one I always find really surprising, and this is the hardest one for students to remember. There's something about the word, people don't remember it. So put a little star by it or something. This one's called transduction. Transduction is a mistake, but because there are so many bajillions of bacteria in the world, and there are so many bajillions of viruses that attack the bacteria, even if it only happens once in a gazillion times, I love these exaggerated words, even if it only happens once in a gazillion times, because there's so many of them, it's actually a significant force to be reckoned with. So transduction is when a bacterial virus, they're called bacteriophages, take DNA from one bacteria and give it to another bacteria. So let's take a look at the diagram here. Oops. Take a look at the diagram here. So here we have a bacteriophage. They're super cool looking. They look like little robots or something. And inside, notice it's a he now, his red DNA. He's going to inject the red DNA into the bacteria's genome. And that in the process of doing that, it causes the bacteria's DNA to disintegrate. The red DNA is his DNA, and it's going to be incorporated into baby bacteriophages. Sorry, wrong phage. However, I want you to notice in stage 3 here, as new viruses are being formed from the DNA, the red DNA, this one accidentally picked up some of the bacteria's DNA. So some of that got incorporated into this particular bacteriophage. So now they explode out of the bacteria, it kills the bacteria, and they go on infecting other bacteria. Only this particular phage has the wrong DNA in it. And so when it goes to inject its DNA, it injects some bacterial DNA. And then that bacterial DNA, they're showing it right here between the dots. So there's dots on it. That bacterial DNA then gets incorporated into the new bacteria. And so this is horizontal gene transfer. We've just gotten DNA from another organism, but not through sexual reproduction. All right, and then the last one for the bacteria, and I'm trying to get my picture. There's no way to get my picture in the right place. This one's probably the most complicated. It's called transposition, and I'm just going to read you a quote straight out of Wikipedia. A transposable element, also called a TE, a transposon, or a jumping gene, all of those are sort of synonyms, is a DNA sequence that can change its position. Sometimes they call it parasitic. It can change its position within a genome, sometimes creating or reversing mutations and altering the cell's genetic identity and genome size. So this is super important. Let me get my picture out of the way here. Let's go with this. In bacteria, transposons often carry genes for antibiotic resistance. They can move from chromosomal DNA to plasmid DNA, which then can be transferred during conjugation, for example, or when the bacteria dies. So these, if you look at these plasmids here, the AMP-R, that is an example, or the TET-R, those are examples of antibiotic resistant genes that are found in the DNA. And here you see it move from the circular DNA. That's sometimes confusing. The large genome of the bacteria is circular, but then it can move to the plasmid, and here you see that it's moved to the plasmid in that circumstance. So that's transposition. 4 ways that bacteria can get new DNA. Transformation, when they suck up DNA from the environment. Conjugation, bacteria sex. Transduction, when a bacteriophage, a virus, transfers the DNA. Or transposition. All right, now the last thing that we're going to talk about is about viruses this time. It's about retroviruses. I thought this diagram was funny. It made me laugh. So when we're talking about retroviruses are called retroviruses because they have a funny flow of information. And I want you to look at this for a moment and think about what's wrong with what you see here. I should have a Jeopardy theme song playing in the background. Do, 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 do. Okay, so what's wrong is that you're not supposed to start with RNA. We always start the genetic code. No, that's, what am I trying to say? The central dogma. There we go. Sorry, brain fart. The central dogma says information flows from DNA to RNA to protein. However, in this circumstance, in retroviruses, we start with RNA. RNA is our initial source of information. It gets converted into DNA and then converted back into RNA and into protein after that. So retroviruses carry an enzyme that is called reverse, let that sink in, reverse transcriptase. So it's going backwards. So instead of going from DNA to RNA, it's going from RNA to DNA. So it reads the viral RNA and converts it to DNA. This is super important. It's a really screwy enzyme that makes all kinds of mistakes. And this is a terrible thing for us as far as creating vaccines, because retroviruses are mutating constantly. I've heard that every single person that is infected with a retrovirus has their own variety of that virus. So every HIV patient has their own special type of HIV. That makes it super hard to create a vaccine against. All right, so I'm going to give you sort of the lowdown on HIV and how HIV works, so that you understand sort of the details of a retrovirus. So this is HIV right here, this diagram right here, and this is a cell that it's going to infect. The cell has all kinds of receptors on the top. You're familiar with that. And so there's one particular receptor that's called the CD4 receptor. This cell is a macrophage. A macrophage is one of the most important cells of the immune system. Its job is to identify invaders, eat invaders, and then sort of start the whole immune system process. All right, so in step number 2, the macrophage has a co-receptor called CCR5, which is this green one right here in the diagram. And so both of them are required to allow HIV to enter the cell. So the macrophages have CCR5 and CD4 receptors. However, T cells also have CD4 receptors, but they don't have CCR5. So initially, HIV infects macrophages only, but not the T cells. In step 3, HIV has an enzyme called reverse transcriptase. So imagine this virus has entered into the cell. It's actually going to enter into the nucleus. It has an enzyme called reverse transcriptase that reads the virus's RNA. So these yellow squiggles, that's RNA. It reads the virus's RNA and makes DNA, its own variety of DNA. But again, it makes a bajillion mutations. So within each patient, you're getting different viral DNA. Number 4, now the viral DNA for any virus, their DNA always says build more viruses, and this is how you build more viruses. So with HIV viruses get made by the macrophage. I want you to understand how insidious this is, because HIV is attacking the very cells that are supposed to be protecting us from it. And yet they've managed to figure out how to invade our immune cells. So the HIV viruses are being made by the macrophage, but they're released by exocytosis. Don't look at this diagram yet, because that's not what's happening. Exocytosis, remember, is when it gets packaged in a vesicle and then released, and that's a normal, healthy part of a way that the cells get rid of things, and it doesn't harm the macrophage. So that's a problem, because now this macrophage can keep producing HIV, and it doesn't get hurt, but it's also not really doing its job. It's not doing what it's supposed to be doing. It's just becoming an HIV virus factory. Some mutant HIV, remember that reverse transcriptase is constantly changing the HIV virus. Sooner or later, some mutant HIV will now require a different co-receptor. So there's a change in the DNA that causes them to need a different co-receptor that's called CXCR4. And it turns out that that co-receptor is only found on T cells. So T cells have CD4 and CXCR4. So they don't get infected initially. This is later on in the infection. When HIV leaves T cells, so the mutant HIV are now entering the T cells. This time, as the T cells start making HIV, they get demolished. So it bursts through the cell membrane. This is the image of it, and it destroys the T cell in the process. An HIV infection is called AIDS, which stands for autoimmune deficiency syndrome, when the CD4 count is below 200. It's normally between 600 and 1500. So that refers to cells that have CD4 receptors. So the fewer and fewer and fewer cells that are patrolling the body, that's when we start saying that this is officially moving from HIV infection into actual AIDS. And then the person who has such a low T cell count or a low CD4 count develops opportunistic infections. So that means infections that our immune system would normally wipe out before we even have a symptom, people come down with them. Things like Kaposi sarcoma, which is a type of cancer, and trying to think of all the different, there's like a type of thrush in the mouth, and there's all kinds of these infections that we wouldn't normally come down with unless our immune system is compromised. All right, cuties, that's it. Let me know if you have any questions.